Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Julia TCW, Assistant Professor at Boston University School of Medicine. She is also the recipient of the 2022 Toffler Scholar Award. Today's discussion is focused on understanding Dr. TCW's efforts using stem cells and improved understanding of genetics to counter Alzheimer's disease. Let's dive in. Welcome, Julia. So nice to have you on this podcast.
Thank you, Deb. It was really, I appreciate the invitation. It's my pleasure to accept this invitation. I'm super excited to discuss about any possible question you might have.
Wonderful. Well, I want to start with, let's just, what is a stem cell? You know, we hear a lot about it. It's in the news sometimes. It can be controversial. But what is a stem cell and why is it a part of your focus of your research?
Okay, it's actually a really good question. Well, we all hear about the stem cell. And actually, everybody has the stem cell. This is a cell that everyone's body that we generate in most of organ. So for example, your hair, nail, skin, when you get a cut, and whichever regenerate, those are called stem cells in the body. So when you're going back, like there are two different kinds, like adult, that's called adult stem cell because we have it and it helps to rejuvenate. But when egg and zygote meet, and that's another type of called and making human body embryonic stem cell. So that's generate a whole body, and that's called embryonic stem cell. And the part that you need to remember maybe is about stem cell property is two things. One is a self-renewal. And the second one is converting them to the various type of cells in a brain organ. So in other words, one property is it keeps replicating the same type of cells. And another property is it has a capacity to generate any type of cells in a body. So this is a beautiful property of stem cell that can be utilized in research because we can make them to go where we want them to go. So for example, if you want to make brain, we can make it from the stem cell. So really fascinating aspect of why we're using stem cell, and especially I study both mouse stem cell and human stem cell. Especially human stem cell is amazing is because all these human diseases such as Alzheimer's disease, and it's a lot of times based on human genetic. And although there are many different type of model from mouse, rodent, different type of rodent, even C. elegans, based worm and fly. However, it's not really well recapitulate our disease. So if we can have our human cells derived from human stem cell that carry human genetic, then we can really mimic what's happening in a human organ or brain that potentially can study human disease.
So you said the stem cells, we have stem cells in every part of our body and that there's a renewal aspect of those stem cells. Do you need brain stem cells to renew brain tissue or brain cells, or can you get those stem cells from somewhere else in the body?
You actually asked super important question. Actually, this is one innovative concept that have received Nobel Prize in 2012, I believe, and about the induced pluripotent stem cell called iPS cell. why is this cell type and making this process become Nobel Prize is because back then we thought that when we different going into final cell type, we cannot revert back to stem cell stage or different type of cells. We thought that it's defined. However, that concept of induced pluripotent stem cell, meaning it actually revert our developmental stage, all this differentiated cell, all the final our skin cell, back to embryonic stem cell. So that is reverting that process, basically rejuvenating back to embryonic stem cell is innovative concept, meaning that actually tell us that these developmental stage can be plastic. It can modulate. It can revert basically progenitor stage, meaning stem cell like cell in a brain can go in neuron, astrocyte, different type of cells. And also embryonic stem cell can also make the whole cell like we expected.
So that's super interesting, and it seems really complicated, I guess, from the novice that I am. But you also study the genetics side of the equation, and I guess what I'm understanding is it's important to understand this kind of going back in time with the stem cells and how you can maybe reset. But where's that relationship to the genetic side? I think I understand it, but can you explain why you have these two focuses when you're studying Alzheimer's?
Yeah, absolutely. So the question to finalize your actual question, basically we can moving back all different cell type, whatever we want by controlling the gene. There we can introduce certain gene. You can make astrocyte to neuron and different type of cells too. So in that perspective, genetic is very interesting component because this gene can turn on certain gene that's supposed to be in one cell type and turn off the other cell type. They change a property. So you can actually manipulate anything you want and convert all these stem cell of even finally differential cell final cell type to another cell type. But what I wanted to emphasize, the reason why we are using the genetic property is that we focus more on our disease stage. And when we look into the genetic, when we compare Alzheimer versus non-Alzheimer patient, and within the Alzheimer, we think that they have a specific mutation they carry. They are prone to develop Alzheimer's disease. Although it is a late onset, well, the familiar form of Alzheimer, such as amyloid, APP gene, presenilin gene, all these familiar type, if they carry, they're 95% become an Alzheimer at early stage. However, these late onsets that people get disease stage or dementia after 65 years old age, and those group of people has particular genetic mutation or variants that they carry that over time accumulate certain effects that potentially lead to Alzheimer's disease. So if we have human cells, which actually derive from stem cell, and we can not only look at their genetic, but also what happened and what cell type, eventually we can identify the mutation effects in cells that we can correct those phenotypes. So hopefully that can be drug development or therapeutic perspective. So that's what direction we are going, so that we think that understanding genetic is very important.
So is it correct in saying that you can, by understanding and seeing how the disease has manifested in late stage people over 65 and knowing what their stem cells may have been, you can basically, and this isn't go back in time to try to understand how the mutation happened or why it happened in that individual?
Oh, let me clarify. So I'm using stem cell tool to make a brain. So you can think that I have your blood and make stem cell and make your brain in a dish. So your brain is, while you're alive, your brain you can see in a dish. Smaller format, but it has all the properties. So that particular cells that is derived from your blood carry all your genetic information. And if while there are limitations that we can see in your brain because you're alive, we can look at what's going on in a dish, your brain. So you have little copy of little organ in a dish. And if something happened in your brain, your probably brain organoid is going to have some problem. So if you can screen a drug with that to fix that particular problem, first of all, we need to identify the problem, right? And then if we can find a solution to fix that issue, then we can develop that drug to treat you. Or for example, if another way is not only drug, if some cell is missing in that dish, your brain in the dish, then we can make that particular cell type and replenish back into your brain with your own cell.
How diverse is each person's brain? If we lined up 10 people and you've created their brain in a dish, how different is each brain? Or is there more similarities than differences?
Actually, you ask amazingly important question. That's why we are studying human stem cells with the human stem cell. It's because we found out that it is so diverse even within the same ethnic background. So in mouse model, a lot of paper published the one mouse strain, and the phenotype is most likely the similar for the same mouse strain. But when we consider same one mouse strain as a one human being, and the next human being is very different, just like a one mouse strain is quite different from another mouse strain. And we thought that if there are same individual, I mean, you know how different we look, right? Even with the same ethnic background, same female, or within same male, everyone looks so different unless you're identical twin. It is, you can see that all those phenotype is so different. Even when we look at global manner, like we tease apart all these genetic and downstream of that one person's organoid versus another person's organoid, it is so different. That's another challenge actually in the field in a research. Like, how could you identify the common aspect? Now, how can we handle these differences? That's actually a big question in the field. And we are trying to develop a way to tackle those.
Because it leads to this idea that there might be only one drug or a handful of drugs that would address Alzheimer's. That's kind of a fallacy. If we're all very different, we may need a very large suite of therapeutics to kind of personalize for each person. Am I understanding that correctly?
Yeah, so it is important question that you're asking and you understood correctly. And another way you can think about is that instead of generating large sets of data and it's in large sets of treatment, and it could be somehow impractical and impact will be less. So instead of that, if we can recategorize these Alzheimer's disease patients, and recently they're working more on early diagnosis using AI, machine learning. And if you can subcategorize not only phenotype, like looking at all their brain imaging or MRIs, or you can also recategorize them based on how many genetic risk factors they carries, or one of the strongest genetic mutation variants that I'm working on, apolipoprotein E, ApoE4, the most risk factor for late onset Alzheimer's disease. You can categorize these patient category, and there are people who are actually going to develop Alzheimer's disease within 2 years versus 10 years. If you subcategorize all these patients, maybe there are people we can target. Let's say if we categorize for 5 different groups and 10 different groups, we can subcategorize that actually can cure this or slow down dramatically before they even get to Alzheimer's disease, dementia, losing memory. So there are a lot of like biomarker work is going on, and people try to now subcategorize this and to better diagnose and to develop better treatment that is appropriate for this group of people. So that's probably the future that we are aiming at.
So it's understanding why the mutations happen in each individual and then categorizing those. And then that will lead to the diagnostic and then the treatment. Is that correct?
Yeah, I think so. That's the ideal way.
Yeah. So we don't really know why the mutations happen, do we?
Well, there are two things. Most of the time, the mutation somatic mutation I was talking about is the mutation that born with it. However, it does not initiate until it get to a bad environment. So when we are young, these type of mutation that inherited, carrying one little tiny nucleotide differences did not affect, and they probably have enough effects when brain is high inflammatory stage or age and something is not functioning right. And then that mutation that carries a particular cell type start reacting. And that's one possibility. And the other case is that when we get age and inflammation happen, and then particular gene is activated, then that should not be activated, actually leading to the bad environment. However, an interesting part is that these days that even if you carries the risk variants, and there are group of people, quite a lot of group of people actually don't develop disease. And people start focusing on those protective side of effects and identify even genetically that there are variants that are protective. And the one example is that we know these familiar cases of Alzheimer's disease. This individual is female individual, carries personality mutation. And usually they develop Alzheimer's disease in around 45 years old, a really early stage, right? Familial disease. But she was healthy until like 77 years old. For 3 decades later, she was still healthy. And we identify this particular variant called Christchurch in ApoE locus. And people have studied what's going on in their brain and that person's brain. And she had this lower level of tau accumulation, although high level of amyloid, somehow protect the brain. So even after that, there are other genetic studies identify multiple other variants that are protective. So maybe if we can play with these protective variants, maybe we can prevent Alzheimer's disease. And that's another direction that we are interested in, as well as others.
Yeah, that's super interesting and hopeful. And I would like to kind of circle back on that a little later in our discussion. But just kind of stepping back here, what inspired you to pursue this career in brain science? And were there any kind of specific moments of influence that ignited this interest that you have?
Yeah, sure. Yeah, for my PhD, I knew that I am interested in understand the mechanism. If I get sick, why am I feeling this way and I feel sick? Rather than, I took a medicine, I'm fine. Not like that. I want to know, why am I in a hospital right now? For what, right? Why am I feeling like this? I want to really understand about the biology. And then actually I joined for my PhD at Harvard studying ALS, Lou Gehrig's disease, as also stem cell. And my PhD is really about understanding these stem cell reprogramming, induced pluripotent stem cell, as I mentioned, all those parts. However, thanks to the lab is studying Lou Gehrig's disease, I have educated for 5 years sitting around a lab meeting, hearing about all the ALS and on neuronal activities and all these brain disorders and how they are studying. And then, I was like educated a lot. I was excited. Then actually there was sad news. My grandpa went through this dementia and he was hospitalized for like 5 years or so. And it affected me that I observed him that he started losing memory and he, at some point, he couldn't remember me anymore. And it was a tough time, but then that actually led me to really thinking about like, I really need to study brain. And that led me to actually studying Alzheimer's disease. And I go check actually my genetic right away. So, luckily I was not carrying E4 variants, but like that was the moment that I want to figure out and the brain, how important the brain health is. And we all get age. And if there's no treatment, that is devastating. And that's another thing, like, the field has been last 15, 20 plus year working on all these neurodegenerative diseases, including Alzheimer, Parkinson, ALS, and all those type of diseases.
Yeah. Yeah. I'm sorry to hear about your grandfather. My grandfather also passed away with dementia, and that it's very difficult for the family. The person just kind of disappears. At least that was the case with my grandfather. He was still there physically, but he just kind of disappeared. His essence just kind of disappeared.
Exactly. I'm sorry to hear that. Yeah, I experienced the same. So I'm like, yeah, we have to fix that because my grandma was very healthy. And that the differences between that, when my grandma passed away too, but the difference between that, until the end of the moment, that how much she communicated with me in fine detail of every little parts, whereas grandpa couldn't. So, yeah. So I think, we need to study the brain better and, find a way to stop this process.
And you said something interesting about you went and got tested to see if you carried the ApoE4. Is that something, are those tests that just anybody can get? Or was that because you were in a lab and you were working science? Is this something that those listeners out there that are curious, they can ask their doctors to test for?
Well, that you actually asked me the very important question. Actually, yes, I actually tested because myself at the lab because I'm working on it all the time. Actually, it's very simple. You just need a saliva. That's it. So I just like put in my saliva in a tube and test it real quick. However, what can we do in public? Good question. For example, 23andMe, you can get, this is a common variant. Everybody can identify the ApoE variant. And if you have ApoE4, having one allele increase the risk for 3.5 fold, and carrying two allele from both mom and pop, you can end up having like 15 times higher chance to get the Alzheimer's disease compared to other. And Asian case, 33 times higher chance. So, whereas it's another interesting point we can discuss later, but African American, Hispanic has much lower level of risk, although they have a pretty high frequency. So, I detour, but coming back. So, 23andMe, you can identify ApoE variant effects, or when you are in an age that insurance cover to test any genetics for the pregnancies, or if you have other inherited genetic risk, probably your insurance is going to cover. But otherwise, probably although in a lab wise, it's easy, but to pay getting service, it could be costly.
Yeah. Yeah. So maybe someday that'd be just a routine that that people, especially as they're younger and healthier, to maybe do. So you mentioned the ethical or the background of people that different kind of races or different kind of backgrounds, genetic backgrounds have different propensity, if you would, to get these diseases. Do we know why?
That's a nature of human genetic. It's a good question. Yes, absolutely. We know to understand. We know why. We need to study more about it. And I encourage actually the people who are actually there are quite a lot of study of European and Asian background of genetic study, whereas much less in Hispanic and African American in the States. And just donate the gene from saliva or anything, and if we can study a lot more of different race, and we can identify each different genetic race could have a different effects on different mutation variants they can identify in different at different ethnic background, and it can cross helping each other. Like if there are certain variants that discover in African American that are actually make the ApoE4 variant protective, we can apply it to European background, or Asian background, which has a higher risk, or vice versa. So if we can study various race of genetic, we can identify protective variants that can help each other.
That's another reason why your lab is looking at those protective variants and that interplay, if you would, with some of the other mutations.
Exactly.
That's interesting. So you talked a little bit about some of the challenges that you encounter in day to day with the work that you're doing, and I also know that you had a kind of a breakthrough. I think it was last year. Some of the work that you were doing with some of the funds from the Karen Toffler Charitable Trust allowed you to do some unique work that you were able to publish on, which led to further funds from NIH, some good funds from NIH. Can you talk a little bit about that breakthrough or that finding, that new knowledge that you discovered in that work? Because it seems to be still pretty fresh, since it's only been about a year or so since that time.
Yeah, thank you for acknowledging that and appreciate it. And also appreciate the charitable trust, the Karen Toffler Charitable Trust. The gene called apolipoprotein E, we just mentioned it for half an hour about it. That's the most risk variants. Variant is another word, easy word is a mutation. Well, it's more penetration when it comes to mutation, but you can think that there's one SNP DNA code mutation causes Alzheimer's disease risk in a very high chance. As I have been mentioning, European case has up to 15 times higher fold increased risk for Alzheimer's disease. So ApoE was discovered in 1990s, and it's been studied a long time with the mouse model and human brain, and identified that this is very complicated gene because it's a lipoprotein, apolipoprotein, meaning you heard about lipids, right? Brain is full of lipids. And of course it has a full of lipids, and this protein carries lipids. It made it from astrocyte and it moved around everywhere in the brain. And we don't really know what this one is doing. And we have been studying a long time. People in the research field have been studying a long time. There are huge genetic association, as I mentioned earlier. And also it actually identified it with the amyloid plaque. So if you carry E4, you have a higher chance to have an amyloid plaque accumulation. Maybe it could be because of ApoE4, or it could be side effects. There are many different things that ApoE4 is doing. Oh, by the way, there are 3 isoforms, 4, 3, and 2. 4 is a risk, 3 is a neutral, and 2 is a protective. And people who carry 4 get Alzheimer's disease easily. The research is about while it's been studied so much, both mouse model, when I first entered the ApoE, it does too many things. And field has been ignored quite a lot is because it hitting too many problem in a E4, and this is not good drug target and also it's a metabolic gene, so it can mess up everything, and we might not be able to cure it. So we study ApoE4, but people are not thinking about targeting ApoE. But what we did in this study is that, well, there are so many different things happening in E4, and I want to figure out what is a major thing that it messed it up, and we need to pay attention, especially human. Well, we will be able to cure the Alzheimer's disease with mouse model long time ago. Amyloid clear up and everything is clean, so humans should be fine. But it's not the case, unfortunately. So what we did in this study is that we compare all these different brain cell type, including astrocyte, neuron, endothelial cell, microglia, and also mixed culture as well of a mimicking human brain from human iPS model with the multiple individual. And there are field controversy is what stem cell model should we use? And we use both population level, we collect it from the multiple individual, as well as we generate almost 4 twins with the giant number of sample. And also we have utilized a large brain bank. In nation, we have Alzheimer's Disease Research Center throughout the country, and they are collecting blood, brain tissue, everything for Alzheimer's patients who are in the study. So we have all those data sets, and also we utilize mouse model too that everybody's using, and compare all those things using computational method called bioinformatics and genetics. And we identify, I will say, as a note, that computer is my best technician. So what we identify is the 2 major dysregulation that's called collateral issue in a brain, and another one pathway called matrisome that's been ignored in the field. It's been identified from the cancer. And these 2 major pathway called collateral and matrisome is dysregulated by ApoE4 in a human-specific manner in human cells. So that's what this paper is about, that when this does so many things, the major dysregulation is that 2, and those 2 are the what we need to go after and target. And also, we further investigate how is this lipid is dysregulated in a cell, and what is a major cell type first dysregulated. And we've identified that was astrocytes, because there are so much controversy in a field that, it's a neuron problem, it's a microglia problem, it's an astrocyte problem, it's an endothelial cell problem, it's a parasite problem. Yes. Well, in our model, we didn't use a parasite, but I believe the parasite has an issue. Where is this major dysfunction started from? We believe that this is a lipid metabolic dysregulation. As we say, it's a lipid gene. It messes up something in astrocyte, and it causes a problem in neuron and leading to the neuronal death. So we demonstrate the mechanism. So we are currently working on how we can target further, and what molecule we need to target it, and how we can screen the drug, and can we go for stem cell therapeutics, and can we identify in our animal model that right treatment window, and those areas that we are working on.
So the image I have while you're going through that description is one of almost like a mechanical function. There's gears, there's pulleys, there's things that are coming in, there's things that are going out. It's a very mechanical kind of look inside the cellular structure of the brain. And so the way I understood what you just said is, something happens, the gears stop, the ins and the outs stop, something stops. And you're looking at why is it not functioning, and what is causing this non-function for that cellular structure. to not act correctly or naturally. And is that, and so you've identified what that, you called it dysregulation of inside this structure, why it's just not working versus just identifying that it's not working. You've identified, here's a couple things that are causing, the healthy process of the brain not to work.
Yeah, I mean, I'm glad you mentioned that. I like it. You going so deep. Yes, we did. Not just like, oh, it's not working. I actually demonstrate the mechanism where what's going on in the cells and which process is defected. So maybe we can correct those processes. So I would use an easy way of analogy as we all experienced COVID. We all experienced at the time that there are difficulty in importing items of you purchase, and it had a hard time to order something and then receive it in your house. I'm sure that all these FedEx and every delivery has an issue. So let me use that analogy that if you look at in a cell, like let's say there is a lipid factory is there, and we can keep running the lipid factory and it's making lipids. However, there is not many workforce to deliver that to outside of cells such as neuron or next door. So delivery chain was broken in this ApoE4 case, that there are not many workforce to carry that lipid to export out to the cells. So that process defected, and it's constantly making, because the regulatory system, there's always a system that senses in a cell that you can stop making more products. However, that process even also defected because their sensors are active. However, they store that in a wrong place so that they don't know where they're stored. So they think that they don't have enough lipid in a cell. So they constantly synthesizing more lipids. And even in a case that there's no deliverable mechanism to deliver to another place. So cell has a full of lipids, and that actually phenotype was first discovered when Alois Alzheimer present amyloid and tau phenotype in his hand drawing. He actually also showed that these Nile red staining, which represent intracellular lipid accumulation around the plaque, and the plaques near cells have a full of lipids. And people haven't revisited that much until recently about talking about this ApoE. That phenotype is probably because of ApoE problem. That lipid problem is not resolved. That actually causes, if lipid is not delivered, neurons cannot survive well, and they cause inflammation, and brain become like very active situation, not only glia, but also all these high inflammatory brain, that something has to make it calm down. So it causes eventually neurodegeneration, and that would lead directly to the memory loss.
It's just so interesting that you can look inside our brains and see this and see that function. How important is collaborating and collaboration in research? You've mentioned other people's work. You've mentioned kind of the, even going back decades of something that, somebody wrote or somebody identified. And now people are just starting to study, how important is that collaboration? And, how do you do that? Because you got to get your work done too.
Oh, yeah. Good question. But it is very important to collaborate. In Alzheimer's field, maybe had a really good culture of collaborative environment. Maybe because we haven't cured the disease too long. So now it's like, let's team up together is kind of normal thing. But especially genetic study, yes, we have to collaborate because we can't study with tens of individuals. Like we need 100,000 of individuals. And that's what sometimes, like, if you look at the genetic paper, there are 100 people, 200 people, is it authors? Because like we are collecting so many individuals, so many centers sample to identify genetic variants, identify what could be a risk, and we have to team up to increase the power. That's one thing that we are working together. And when we go back, like, let's talk about the ApoE part. Like, genetic for sure, we got to work together. So we actually do have a weekly meeting, bi-weekly meeting, monthly meeting, even conference to put all these bits of it together to make the work more powerful so that that result can be, you heard about the Manhattan plot, right? The result can be meaningful. It can be translational. It can be applied to actually benefiting human being. Otherwise, one lab cannot do that kind of work. It's just impossible. But let's go back and talk about like ApoE. Well, when you only look at one gene and one protein, do we still need to work together? Absolutely, we need to work together. Still, so many things happening. And there are structural biologists who actually how ApoE is looking different, carrying different mutation. How are they wrap around lipid differently? Well, we don't really know how they're wrapping differently. And proteomics, lipidomics people, animal biologists, clinical pathologists, geneticists, cell molecular biologists, we all got to work together to better understand these important targets for Alzheimer's disease or even other diseases. So one lab contribution could be limited, well, but each lab can contribute and then we all together understand better way. And that's why we think that collaboration is super important. Where is it happening? Well, most of the time we go to conferences and, we go in the same session and talk about the same thing and then in a different angle. And end up like we identify something else. Oh, I need this amazing structural biologist to look into this, where it's binding to. Then they have all the tools to look into how they are binding to receptors. And we have work to like provide our resource to looking in our human cells. And that's how the collaboration happens.
So how important is policy and funding? How does that influence just the landscape of, basic brain science? Is it helpful? Do you need more of it? What does it, where does policy and funding come in to help you progress your research?
Especially the policy part has been dramatically changed, especially for stem cell field, which I really personally feel it, how much it has been changed and it benefiting our research. Like back in when I was at Harvard, when we first studied about the embryonic stem cell, well, I started before even stem cell word is even utilized. There are certain work allowed only in Massachusetts and the more broadly approved in New York City, New York states, and different state has different regulation. But when I remember that I work in New York City for postdoc, I still need to get an approval from Institutional Review Board for the waiver to work with the human iPS cells. But then when I moved into Boston in 2021, now I found out that their even waiver is even waived. So I can just write about what I'm working with and approved by the Institutional Biosafety Committee. It's a part of the safety issue. Nothing harmful, anything. It's just research material. And simply like in last 16 years, it's been changed dramatically. So definitely it benefit research and expand the whole field that more people can jump into this using stem cells as a model to work with the human diseases. Although still there are some limitation between the crossover between academia and industry. But in terms of like a brain the related to policy and funding related to brain research, I think, this philanthropic funding and government funding is more and more to provide a resource to help human disease and also brain and brain health. Because relatively, I heard from the senior PIs that was much smaller fundings in NIA, but these days they're, more people are interested in brain because as it is one of the most difficult to tackle in a body, but it is very important that it controls the whole human body. So understanding brain better and focusing a lot in a disease field that actually policy as a funding source, it should be more expendable. And I hope that more philanthropy contribution and government funding is towards the brain, so we can really live a healthier life.
Do you feel like it's accelerating? Do you feel like the progression of understanding of the brain and the diseases of the brain, are we speeding that process up? Is it accelerating in your perspective?
Oh, absolutely. Yes. But think about like a cancer, their funding was a lot more, and there are way more drugs out compared to the brain, because brain, it was like their barrier is big. They think that it's difficult to, cure it, but even slowing down is dramatic, and more funding actually equivalent to identifying more drug targets and drugs out in the field. So, well, it's only one or two like targeting amyloid exists, but we have to identify more different type of targets and drugs and therapeutics available in the field. That could be the future, and I'm sure that more funding should be made in this field.
So, talking about the future, what do you see as the future of brain science? Where is it heading, and what are you most excited about in any kind of developments or possibilities on the horizon?
Well, with the team effort, we identified and developed we will identify and develop many drugs and therapeutic, not only drug, but also other tools using stem cell therapeutics and or genome editing by CRISPR and so on. So another aspect is that the future, we would use machine learning or AI to identify subcategorizing patients and applying genetics to early diagnose. And, we can identify the potential disease candidates who, we can treat earlier, way earlier, before they even losing any memory or any damage, to cure the Alzheimer's disease. So if we can extend longer than human longevity to prevent it, and then everybody can have a healthy brain. So that's the goal.
So the goal is to have these kind of converging technologies or understanding to where individually, if before we start to mutate into the disease, that we can go back and fix it, basically reboot our genetics to avoid the disease.
Yeah, exactly. That's possible.
That's amazing.
That will be possible in the future.
Yeah, that's amazing. What do you wish people knew about the process of research or what you do on a day-to-day that they may not know? I mean, there's a lot of people out there that may not know researchers. They may not understand the scientific process. What do you wish they knew that maybe they don't know?
Okay. Maybe I can explain about my case. Everyone goes to hospital, and when I ask public about who is making this medicine that your doctor prescribe you or IV you to rescue you, and the majority answer I heard is a pharmaceutical company. Yes, that's the right answer. And I would say research is a process of understanding every pieces of like solving a puzzle. So to make a medicine, we need to first understand our human cell from that forms an organ that connects to be functional. And we need to understand where this dysfunction derived from to correct and to make a medicine. So I actually myself appreciate and get benefit from medicine, that every time when I'm at ER by any chance, and understand how devastated the situation can be if there's no medicine. So imagine that those time, if you've ever been in an ER emergency room, and I would like to understand what and how human body can react on certain situation and certain environment, and identify a target and make a medicine. So there are more medicine, there are way more ways that we can live a better life and not being sick. So I'm happy and proud of myself that I can contribute to the world in this way. And day-to-day life, yes, I am every day trying to solve the puzzle and try to understand the one little pieces of part that matters to us and human body.
In your lab, because you have a lab, you have other researchers that are starting their career or helping some of this science out. Can you describe what it's like to work with these people too?
Yeah. Oh my, yeah, it's really exciting actually. And I'm very excited about like every day motivated them that how important work they're doing. I'm training multiple level of students to the doctors too. People from undergrad, sometimes high school student, college student, master student, PhD student, doctoral degree student, and medical student, MD student, and postdoc who receive already a doctoral degree, who actually still in a training process of called postdoctoral fellows, and even faculties. And also we do a peer mentoring too, so another faculties too. But they are I want them to have a, same page of me every day that how important work they're doing and how much they're contributing in the world and impactful their work is. And I'm sure like most of people in my lab actually get excited and they're just happy to, discover something new and that could be potential therapeutics. So yeah, the sharing that moment and everyday work is hard because a lot of times that we do work with very invisible part that to make it visible and to quantify. So a lot of times in a paper, it's a process of invisible information to be visible so we can analyze and find the issues and identify even correct how we can correct those and that can potentially hopefully one day we make a medicine and going through clinical trial hopefully it can be in a shelf and you can have it when you're sick.
So you talked about collaboration being very important, that scientists and researchers all over the world kind of building upon each other's information. How would people that are listening to this, how would they get a hold of you if they wanted to collaborate or if they just had questions about what you're doing and what you're learning? How would people get a hold of you?
It's very simple. You can just email me. My name is Julia TCW. So you can type juliatcw@bu.edu, or you can connect me with LinkedIn. You can just Google my name, Julia TCW, or TCW Lab. You can see what we are doing, publication, contact, and or in public, drtcw@gmail.com. Feel free to contact me. So any way you would like to contribute, and even you want to make your own stem cell, there are ways to do it. And our team can get a consent, and then you can actually have your own stem cell as well. So please feel free to contact me for collaboration, any further information. Then yeah, I'll be appreciate for your participation, and it'll be exciting.
Yeah, Julia, this has been really interesting. What you're working on is just, it's for the layperson, it's very hard to understand. So thank goodness for your understanding and your dedication. But to peer inside the cellular structure and kind of the mechanics of what's happening in the brain, and then to bring that side of the equation around the stem cell and some of the other technologies that you talked about, the computational and the AI to look at not only what's happening, but why is it happening, so that we can someday maybe have therapeutics that make Alzheimer's not existent, or possibly even reboot ourselves so that we don't get these mutations. That's an amazing kind of future to think about. So I really appreciate your time today. It's been wonderful. We've known each other for a few years now. And every time I talk to you, it's wonderful. I feel your passion and your excitement for the future. So thank you very much for spending your time today.
Thank you very much, Deborah, for your invitation. It was my pleasure.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.